Hello and welcome to the cell cycle lesson. From your previous work, you already know about chromatin and how it is spread out in a eukaryotic cell that is not dividing. Sexually reproducing organisms get half of their chromosomes from one parent and half from the other. The chromosomes donated by one parent make up a complete set of genes required for an organism. So you actually have two copies of each gene, one copy from one parent and one copy from the other parent. The chromosomes containing the same genes are known as homologous pairs. They are the same size, carry the same genes in the same locations. While the chromosomes in the homologous pair have the same genes, the form of the gene may be different. For example, the gene located here in this region may be the gene for hair color. The maternal, or the chromosome that came from the mother, may be have the code for blonde hair, while the paternal chromosome, the chromosome coming from the father, may have the code for brown hair. When a cell has both sets of chromosomes, it is referred to as diploid. It has two of each type of chromosome or contains the homologous pairs. If a cell only has one set, as in human egg cells or sperm cells, then it is referred to as haploid. Now that we've talked about chromosomes and homologous pairs, we can begin our discussion of the cell cycle. The cell cycle is the life cycle of the cell and has various stages. The first stage is interphase, and it is the longest stage of the cell cycle. Interphase is divided into three stages. The first is G1. A new cell is formed, and it is half the size of the original cell. It grows until it reaches maturity and then begins carrying on the normal functions for that cell. Many cells spend their whole lives in this stage. So if that is the case, for them, we refer to them as being in G0. If a cell gets a signal to divide, it will enter the S stage where it makes a copy of the DNA. You will remember that DNA is copied in a semi-conservative method. The DNA is unzipped and a new strand is built, matching with the old strands on both sides. So the two new copies are half original DNA and half new. When the DNA has been copied, the cell then enters the third stage of interphase, which is G2, which is preparation for division. After the DNA is copied in the S stage, it is now referred to as a duplicated chromosome and consists of two sister chromatids held together at the centromere. That is the constricted point right here that you saw before on the individual chromosomes. Well, that's the point at which the two copies are attached together. This is not a homologous pair. This is one homolog with its copy attached. Proteins, known as kinetochore proteins, are also located at the centromere, and these proteins attach microtubules to the centromere. Here is an image of the complete cell cycle. Just as a review, we want to see how interphase is divided into G1, S, and G2. G1 is the growth stage. S is where the copies are made, so the two sister chromatids are attached together. And then in G2 you have the preparations for actual division. The next stage is mitosis. And mitosis is divided into, in this picture, 5 stages. But I usually combine late prophase with prophase, and so I prefer to just remember 4 stages: prophase, metaphase, anaphase, and telophase. After mitosis, the final stage of cell division is cytokinesis. Students often link it with mitosis, but it is a separate stage. The definition of mitosis is nuclear replication. You're making a copy of the nucleus. So you've copied the DNA. Now you're going through the steps to separate the two copies into two new nuclei. The first step is prophase. There are four major things going on here. The nuclear envelope and nucleoli have to be disassembled. The centrosomes have copied themselves and they move to opposite poles or opposite ends of the cell. And spindle fibers made of microtubules will begin to appear. Chromatin condenses down into those X-shaped duplicated chromosomes we talked about, and the centromeres get attached to the spindle fibers. This is an image of early prophase. The chromosomes are condensing, the nuclear envelope's beginning to disappear, the centrosomes with centrioles are moving to opposite poles. So this is an animal cell, so they're separating, moving this way and this way. And microtubules or spindle fibers are beginning to arise from those centrosomes. This is an image of late prophase, where the duplicated chromosomes have completely condensed and they are attached to spindle fibers, and the centrosomes with their centrioles are on opposite poles. After prophase, the next stage is metaphase. The chromosomes are moved by the microtubules to line up on what's referred to as the metaphase plate. It is a plane through the center or equator of the cell. Under the microscope, it often looks like they are in a straight line. But when you look at it from an angle, you'll notice that they are actually arranged in a circular pattern along that metaphase plate. The next stage is anaphase. The sister chromatids, or copies, are separated and pulled to opposite poles. These copies are now referred to as daughter chromosomes. The last stage of mitosis is telophase. The sister chromatids have been pulled apart from each other and now at opposite poles. They will decondense back into chromatin. Nuclear envelopes will form around each set of DNA, and the spindle fibers will disassemble. There is now two identical nuclei in one cell. Mitosis is now complete. The cell may or may not go through cytokinesis, and cytokinesis may overlap with telophase. Cytokinesis in animal cells begins at the metaphase plate when a contractile ring of actin filaments begins to squeeze, pinching the cell in half. This creates a furrow known as the cleavage furrow. The contractile ring continues to squeeze, pinching the cell in two. Sometimes it can start at one end of the cell and just squeeze till it gets to the other side, sort of slicing the cell in half. Cytokinesis in plant cells happen when vesicles with cell wall material travel to the metaphase plate and fuse midway between the daughter nuclei. It is then called a cell plate. So here you can see the cell plate forming where the vesicles are fusing together. In the close-up on the right, you can see where vesicles are coming into contact with each other. Their membranes will fuse together, connecting the cell wall material inside into a new cell wall that will divide the cell into two new cells, and the membrane portion of the vesicles become the new plasma membrane, one on this cell and one on this cell, and they will merge with the existing plasma membrane. If I can get my stylus to work, and the cell wall material will fuse with the existing cell wall, thus dividing the cell into two new cells. It is easier to observe plant cell mitosis compared to animal cell mitosis due to the cell walls holding the cells in nice, orderly fashion. Typically in a general biology class, onion root tips are used to observe mitosis because they are growing rapidly, so their cells are undergoing mitosis. The root tips are sliced thinly, fixed and stained, and placed on microscope slides. They appear with a darker region where the cells are dividing rapidly. Under higher magnification, you can see individual cells. Here, we have a cell in interphase. You can see a nice round nucleus and even a nucleoli. Here you can see the other stages of mitosis. This first cell is in prophase. Here we have metaphase. Anaphase is down here where the sister chromatids are being pulled apart. And telophase, where you can see a cell plate forming, which is also cytokinesis overlapping with telophase. Observing animal cell mitosis is done using eggs from a whitefish. The eggs are in a stage known as the blastula stage, which is essentially a hollow ball of rapidly dividing cells. The blastulas are fixed and stained and sliced thin and placed on microscope slides. So here you see the slices of the fish eggs. Under higher magnification, you can see cells in the different stages of mitosis. Here is one in interphase. Here are whitefish blastula cells going through mitosis. Prophase, metaphase, anaphase, telophase, and cytokinesis, and here is your cleavage furrow. In summary, a cell spends most of its time in G1. Some never leave, so we refer to them as being in G0. If a cell gets a signal to divide, it will then enter S stage and make a copy of the DNA, prep in G2 for nuclear replication, go through mitosis, prophase, metaphase, and anaphase, and telophase, and may or may not then go through cytokinesis to form two new cells that are half the size of the original, and the process starts all over again.